Welcome to Eduimagica, the medical mystery education hub. Today, we are going to learn about electron transport chain. Now, before watching the video on electron transport chain, also make sure to watch videos on glycolysis, Krebs cycle, and beta oxidation for better understanding. Because these cell pathways are linked to each other in one way or the other. You can find the links for the videos in the description below. Before beginning, make sure to subscribe our channel and hit the bell icon so you don't miss any future updates. Now let's talk about the overview of electron transport chain. Electron transport chain is a set or chain of enzymes that are located in inner mitochondrial membrane, and we know that it contains 4 enzyme complexes located in the inner membrane. Electron transport chain is main source of energy in aerobic pathway of energy generation. Now let's talk about the structure of mitochondria. The outer membrane of the mitochondria is known as outer mitochondrial membrane. The inner membrane is known as inner mitochondrial membrane, and the folds of the inner mitochondrial membrane is known as cristae. And inside the inner mitochondrial membrane we have matrix, and the space between the outer and the inner membrane is intermembrane space. Now let's talk about the structure of the membrane of mitochondria in detail. Now here we have outer mitochondrial membrane, we have inner mitochondrial membrane, and in between them we have intermembrane space. And we know that in inner mitochondrial membrane we have 4 enzyme complexes, and these enzyme complexes are numbered from 1 to 4 in Roman numerals. And inside the inner mitochondrial membrane we have matrix, and matrix of mitochondria also contains other cycle such as Krebs cycle and beta oxidation. And we know these cycles produce reducing equivalents NADH and FADH2. These NADH and FADH2 contains electrons that are donated to the set or chain of enzymes complexes. The electron then travel from the first to the fourth enzyme complex, and in this movement energy is generated. This energy is utilized in pumping hydrogen ions from matrix to intermembrane space. This continuous pumping of hydrogen ions in the intermembrane space results in generation of higher concentration of hydrogen ions in the intermembrane space. This generates positive charge in the intermembrane space and a negative charge in the matrix of mitochondria. This is known as electrochemical gradient. The hydrogen ions cannot cross along this electrochemical gradient because the inner mitochondrial membrane is impermeable to ions. So here we require a special transporter. The enzyme ATP synthetase comes into play. This pumps hydrogen ions into the matrix and uses the energy generated from the flow of hydrogen ions to phosphorylate adenosine diphosphate to adenosine triphosphate. This is the basics of energy production from the electron transport chain. Now let's talk about the different complexes of electron transport chain. As we all know, we have outer mitochondrial membrane, we have inner mitochondrial membrane, and in between them we have intermembrane space. And in the inner mitochondrial membrane we have 4 enzyme complexes, and these 4 enzyme complexes are numbered from 1 to 4 in Roman numerals. And we know that along with it 4 enzyme complexes we have 2 prosthetic groups. These are coenzyme Q and the cytochrome C. The cytochrome C works as the intermediate between the third and the fourth in transport of electrons. Now let's talk about the complex number 1. The very first complex is also known as NADH dehydrogenase complex and or NADH oxidoreductase. Now this is very important. Why it is known as NADH dehydrogenase complex? It is so because it is a dehydrogenase which removes hydrogen from reduced form of nicotinamide adenine dinucleotide. It is known as complex because it also contains FMN and iron sulfur compounds. Now why it is known as NADH oxidoreductase? Because dehydrogenation is an example of oxidation reduction reaction. The main role of this complex is to receive electron from NADH and transport further into the electron transport chain. The complex 1 is a L-shaped protein complex. And this complex has two arms, the vertical and the horizontal arm. The vertical arm is embedded in the inner mitochondrial membrane, whereas horizontal arm is embedded in the matrix of the mitochondria. In this process, NADH donates its electron to the complex 1, which further transport the electron into the electron transport chain for the production of energy. Now let's talk about the complex number 2. The complex 2 is also known as succinate dehydrogenase complex. Now here also the question arises why it is known as succinate dehydrogenase complex. It is so because it is a dehydrogenase which removes hydrogen from succinate and oxidizes it to fumarate, which is a step of Krebs cycle. And similarly, it is known as complex because it contains iron sulfur clusters and succinate dehydrogenase. The second complex is a part of Krebs cycle which catalyzes the reaction for oxidation of succinate to fumarate. And during this reaction, the reducing equivalent FADH2 is produced. And this reducing equivalent FADH2 donates its electron to the complex number 2, which further donates its electron to coenzyme Q or the ubiquinone. Now let's talk about the complex number 3. The complex number 3 is also known as cytochrome reductase or Q-cytochrome C oxidoreductase. Cytochromes are the group of proteins which have heme as their complexes. They also have iron core in which iron can exist in oxidized or the reduced form, depending on to the donation or receiving of the electrons. In the third complex, we have cytochrome C1, cytochrome B, and we have cytochrome C. The electrons are first received by cytochrome C1 and cytochrome B, and the electrons further get transported to cytochrome C, and from there it gets further transported to the fourth complex. Now let's talk about the complex number 4. The complex number 4 is also known as cytochrome C oxidase. This complex contains hemes and copper units. Its main job is to oxidize cytochrome C. The cytochrome C donates its electron to complex number 4. And during this process, the electrons are accepted by the oxygen and formation of the water takes place. Now let's talk about the reactions in details. Now here we have the outer mitochondrial membrane, the intermembrane space, and the inner mitochondrial membrane. Now here is an illustration that resembles the structure of mitochondrial membrane as well as organization of ETC complexes. In matrix of Krebs cycle, we have various biochemical cycles such as Krebs cycle and beta oxidation. These generate reducing equivalents NADH and FADH2. The electron transport begins when the reducing equivalents which is produced from the Krebs cycle and the beta oxidation donates its electron to the first complex. The electron in the illustration is marked with the red color. In the first complex, the electron is first received by the flavine mononucleotide, which is a prostatic group. Then it donates its electron to the iron sulfur proteins. These are the redox reaction in which iron ions go from ferrous state to the ferric state, depending on donation and receiving of electrons. In the next step, the iron sulfur protein donates its electron to coenzyme Q. Q is a ubiquinone. which is a freely permeable molecule inside the inner mitochondrial membrane. These redox reactions generate energy that is utilized to pump 4 hydrogen ions from matrix to intermembrane space. The coenzyme Q then combines with 2 hydrogen ions from matrix of mitochondria and get converted into reduced form of ubiquinone. Now coming to the second complex, we have learned about it earlier that this complex is involved in Krebs cycle and contains an enzyme known as succinate dehydrogenase. And the FADH2 produced donates its electron to the iron sulfur complex of complex number 2. The electrons are then transferred to ubiquinone. The ubiquinone is freely permeable molecule inside the inner mitochondrial membrane, which is used to transfer electron to the third complex. In the third complex, the electron is first received by cytochrome C1. Then the electrons are transferred to the iron sulfur protein, which then donates its electron to cytochrome B. These are all redox reactions. Then the electrons are transferred to cytochrome C, which is an intermediate protein used to donate electron to fourth complex. And during this process, the energy generated is utilized to pump 4 hydrogen ions from matrix to intermembrane space. Now in the next step, the cytochrome C donates its electron to fourth complex. In fourth complex, the electrons are first received by copper ions of A type. Then it donates the electron to cytochrome A. Then further to the copper ions of B type. And in the final step, it is donated to cytochrome A3. The cytochrome A3 in the final step donates the electron to the oxygen, which is the final acceptor. The oxygen combines with 2 hydrogen ions from the matrix of mitochondria and gets reduced to water. This redox reaction causes pumping of 2 hydrogen ions from matrix to intermembrane space. All this pumping of hydrogen ions in intermembrane space causes the generation of higher concentration of hydrogen ions in intermembrane space as compared to matrix. Inner mitochondrial membrane is impermeable to ions, so ions cannot cross membrane as such. So we require a special enzyme complex, that is ATP synthetase. These ATP synthetase pumps 4 hydrogen ions and the energy generated by it is utilized to phosphorylate ADP to ATP, and thus the energy is produced in the form of ATP. Now let's talk about the energy calculation. For the energy calculation, here we will calculate the energy for the per molecule of glucose. For this, we will look into the different cycles, the glycolysis, Krebs cycle and the electron transport chain. In the glycolysis, when the glucose gets converted into pyruvate, it generates 2 molecules of ATP and 2 molecules of NADH. And when the pyruvate gets converted into acetyl-CoA, it generates 2 molecules of NADH. And in the Krebs cycle, we get 2 molecules of GTPs, which is equivalent to 2 molecules of ATPs, 6 molecules of NADH and 2 molecules of FADH2. So in electron transport chain, we have 2 NADH, 2 NADH, 6 NADH and 2 FADH2. And 2 NADH is equivalent to 5 ATP. 2 NADH is equal to 5 ATP, 6 NADH is equal to 15 ATP and 2 FADH2 is equal to 3 ATP. So in total we get 2 ATPs from the electron transport chain. So here 28 ATPs is from the electron transport chain, 2 ATPs or 2 GTPs from the Krebs cycle and 2 ATPs from the glycolysis. So in total we get 32 molecules of ATP per glucose. Thank you for watching. You can follow us on Facebook, Instagram and Twitter. The links are in description below. You can also visit us at www.eduimagica.com. Please hit the subscribe button and follow us for more.